Good morning to Mr. Ayman and everyone. My name is Shrikant, so I'm from Diploma MLT. Course code and name will be MLD 3213 Transfusion Science, which is for the semester April 2026. So today I will be presenting on antiglobulin testing, the cornerstone of the transfusion science. So in this presentation, I will explain the principle of the antiglobulin testing, the two testing pathways, and also the importance in laboratory diagnosis and transfusion science. So, without wasting time, let's jump into the topic. The diagnostic challenge related to the transfusion sites. So, in transfusion times, clinically significant unexpected antibody can coat red blood cells and cause severe life-threatening destruction, which also known as the hemolysis. However, the problem is that red blood cells coated with IgG antibodies, which is the immunoglobulin G antibodies, do not agglutinate directly. Because of this, the reaction is invisible to the naked eye. Kita tak boleh nampak dengan mata kasar. Therefore, we need a way to build a bridge so these hidden antibodies can be detected. So, untuk itu yang kita pakai antiglobulin testing. So, the antiglobulin testing is also known as the Coombs test. So, the core purpose of the antiglobulin test, which is the primary function, is to detect clinically significant unexpected antibodies that have coated red blood cells. So, the test can detect in vivo coating, which means the antibodies attach inside the body. So, the reaction occur within the body, and in vitro coating, which refers to antibodies that attach to red blood cells inside a test tube, which is outside the body during a laboratory testing. So for the AHG testing, kita pakai solution AHG, which is the anti-human globulin solution ataupun the reagent, which bridging the gap. So in this slide, I'm going to explain about how the AHG works. So in phase 1, the sensitized cells are present, but there is no visible agglutination or the clumping of the red blood cells. In the phase 2, the AHG reagent is added to the mixture. Finally, in the phase 3, the visible agglutination occurs. This is because the anti-human globulin creates a physical bridge between the isolated antigen-antibody complexes, transforming the invisible cellular coating into a visible agglutination. So the agglutination can be observed in two ways. First is the macroscopic agglutination, where visible to the naked eye. The second is microscopic agglutination, which only can be seen under a magnification using the microscope. So both observations help the laboratory professionals to determine whether the test result is positive or negative. So that's all for the principles of the antiglobulin testing. Let's go for the next, which is the two pathways that involved in the antiglobulin test. So the first pathway, which will be the direct antiglobulin test or the DAT. It detects antibodies or complement that coating the patient's cells in vivo, meaning inside the body. The second is the indirect antiglobulin test, which or IAT. It detects antibodies or complement coating the patient's cells in vitro, meaning inside the laboratory using a test tube, which involves the patient's serum and also the donor's red blood cells. So let's compare the direct and also indirect antiglobulin test. So in this table, we can differentiate both of them. So for the direct antiglobulin test, the starting sample is the patient's washed red blood cells. So the detection target is antibodies or complement that coated the red blood cells in vivo, which is inside the living body. Its clinical goal is to diagnose active immune-mediated hemolysis. For the indirect antiglobulin test, the starting sample is the patient's serum together with the donor's red blood cell. The detection target is antibodies or complement coated red blood cells in vitro, which means inside the laboratory, outside the living human body. Its clinical goal is to screen or prevent future incompatible transfusions or pregnancies. So this slide illustrates the principles of both direct Coombs test, which is the direct antiglobulin test, and the indirect Coombs test, which is the indirect antiglobulin test. So let's begin with the direct Coombs test, which is shown at the top. So the first, a blood sample is collected from a patient with the immune-mediated hemolytic anemia. So the red blood cells already have immune antibodies attached to the antigens on their surface. So the patient's washed red blood cells are then incubated with the anti-human antibodies, which is the ASG reagent. If the human antibodies are present on the red blood cells, the anti-human globulin bind to them and link the red blood cells together, producing a visible agglutination, which indicates a positive test result. So now look at the indirect Coombs test, which is at the bottom. So the first, the recipient's serum is obtained, which contains the immunoglobulins or the immunoglobulin G antibodies. A donor's blood sample is then added to the tube containing the recipient's serum. If antibodies are present, the recipient's immunoglobulin G binds to the donor's red blood cells and form an antigen-antibody complexes. After that, the anti-human globulin or the Coombs antibodies are added. So these antibodies bind to the human IgG attached to the donor red blood cells, causing the agglutination. So the visible agglutination represents a positive indirect Coombs test result. So the flowchart explains about the standardized laboratory procedure. So the direct antiglobulin test follows four standardized steps, where the step 1 is the antigen-antibody reaction, where the red blood cells are already coated with the IgG antibodies or complement proteins inside the body, a patient's body. Step 2 is the crucial washing step. So the patient's red blood cells are washed several times, which is about 3 times with normal saline solution to remove any unbound or free antibodies or complement proteins. Step 3 is the addition of AHG reagent. So the anti-human globulin reagent binds to the antibodies already attached to the red blood cells and bridges them together. So the step 4 is the quality control, which using the Coombs control check cells, also known as the triple C, to validate the negative results of the AHT reaction and also confirm that the AHT reagent is working correctly. So let's go detailed about the principles of the two pathways. So direct antiglobulin test, which is detecting in vivo sensitization. So this slide explains the principle of the direct antiglobulin test. So the direct antiglobulin test detect red blood cells that have been sensitized with immunoglobulin G antibodies, complement proteins, or both inside the patient's body, which is known as the in vivo sensitization. So in this diagram, we can see the immunoglobulin G antibodies and complement proteins already attached to the patient's red blood cells before the test is performed. So the purpose of the direct antiglobulin testing is to confirm whether the immune coating has already occurred in the patient's circulation or not yet. So the direct antiglobulin test detecting the in vivo threat. How? They detect the red blood cells that have been sensitized with IgG, autoantibodies, or complement proteins inside the patient's body period to the blood draw. The positive direct antiglobulin test can be seen in several clinical conditions. So what is the triggers of immune-mediated hemolytic anemia? So the first is alloimmune hemolysis, which includes the hemolytic transfusion reactions and hemolytic disease of the newborn. The second is autoimmune hemolysis, which including the autoimmune hemolytic anemia, where the antibodies react against the patient's own red blood cells. So, for example, kita ada warm antibodies are IgG, which involved in the SLE, systemic lupus erythematosus, and also the Evans syndrome. And then cold antibodies such as the IgM, which involved in the infectious mononucleosis and also the paroxysmal cold hemoglobinuria. And the third is the drug-induced hemolysis, where certain medications such as the penicillin or the methyl dopa can trigger the immune-mediated destruction of the red blood cells. So this slide explains about the direct antiglobulin test laboratory workflow. So I already explained about it. It's just a summarize of that. So I just skip this and go for the next slide. Next, let's talk about the indirect antiglobulin test, which is detecting in vitro sensitization. So the indirect antiglobulin test detects unexpected antibodies which present in the patient's serum. Unlike the direct antiglobulin test, the antibodies are not yet attached to the red blood cells. So the patient serum is mixed with the reagent red blood cells and incubated to allow antibody sensitization. After incubation, the cells are washed to remove free or unbound antibodies or the complement proteins. So the AHT reagent is then added. If agglutination occurs, the result is positive, indicating the presence of clinically significant antibodies. So what is the clinical uses of the indirect Coombs test ataupun the indirect antiglobulin test? So the indirect antiglobulin test has three important clinical applications. So the first application is the antibody screening and identification, which detects the clinically significant unexpected antibodies in the patient serum. The second is the cross-matching, where the donor blood and also the patient blood are tested for compatibility before transfusion. The third application is the antenatal screening, where pregnant women are screened for immunoglobulin G antibodies that could cross the placenta and cause hemolytic disease of the newborn. So now let's see the indirect antiglobulin test laboratory workflow. So this slide illustrates the complete indirect antiglobulin test workflow. So first, the patient serum is collected. The serum is then incubated with reagent red blood cells, which is known red blood cell groups. And then the cells are then washed several times to remove the unbound antibodies or the free complement proteins. And then the AHG reagent is added. So finally, the sample is examined for the agglutination. So agglutination occurs, which means indicates a positive result, while the absence of agglutination indicates a negative result. So now let's see about the AHG, which is the Coombs reagent ataupun the anti-human globulin. What is it? So the anti-human globulin, which is the reagent, also called Coombs reagent, is the key reagent used in both direct and also the indirect antiglobulin tests. It is produced by injecting human globulin into the animals to stimulate antibody production. So the reagent actually contains antibodies against human immunoglobulin G and in some formulations, antibodies against complement proteins as well. So its function is to bind to sensitized red blood cells and produce a visible agglutination. So now let's talk about the quality control, which is the Coombs control check cell verification. So the Coombs control check cell verification is an important quality control procedure. It is performed only after a negative antiglobulin test result is yield. So the Coombs check cells are the IgG coated red blood cells that are added to every negative result from the AHG test. So the CCC are adalah group O positive cells that sensitized with the anti-D. So if agglutination occurs after adding the check cells means it confirms that the AHG reagent was active and then the washing procedure was adequate. However, if there is no agglutination after adding the check cells, the test is considered invalid and must be repeated again. So the physics of repulsion, which is the zeta potential. So the red blood cells naturally carry a negative surface charge ataupun a negative net charge on their surface. So this creates a force known as the zeta potential, which causes the red blood cells to repel each other. So because of this repulsion, the IgG antibodies alone are usually unable to produce a visible agglutination. So the anti-human globulin reagent overcomes this problem by bridging the sensitized red blood cells together, which allowing visible agglutination to occur. So to overcome this repulsion, there is enhancement media, which is also known as the potentiators. So the potentiators are the reagents that are used to reduce the zeta potential between the red blood cell membrane. So their purpose is to make the antibody detection faster and more efficient. So the common potentiators that are used include LIS, which is the low ionic strength solution, albumin, polyethylene glycol, and proteolytic enzymes. So these reagents improve the sensitivity of the antiglobulin testing by enhancing the antigen antibody reaction. So, and we have arrived to the conclusion part. To summarize, the antiglobulin testing reveals the hidden world of the sensitized red blood cells by using the anti-human globulin reagent to breach the immunoglobulin G-coated cells and produce visible agglutination. So the direct antiglobulin test detects the antibodies attached to red blood cells inside the patient's body, while the indirect antiglobulin test detects antibodies present in the patient's serum before they attach to the red blood cells. Together, these tests are fundamental tools in transfusion science because they help ensure safe blood transfusion, diagnose immune-mediated hemolysis, and protect the patients from serious transfusion complications. So far, these are the references I have used for my presentation assignment. Thank you for listening to me. That's all from me. Thank you.